Welcome to the Bio Guys Do the Bio IA. In this video, we will be going over the analysis section. Mr. Brown, many students struggle with the analysis section. What are some difficult parts of the analysis that students oftentimes struggle with?
Well, the analysis section contains many different subsections, each with their own requirements. I think properly completing each subsection can be overwhelming and difficult, not to mention the fact that the actual statistical analysis itself can be difficult to complete.
Absolutely. The analysis section can look a little overwhelming because there's so many different aspects you have to keep track as you go through this section. So we'll just be going over the analysis section by section, using the score sheet, look at what you need to do to get a 7 on this section. And keep in mind, if you do well and get a 7 on this section, you're set up fantastically for overall throughout the Bio AA, and you should be well on your way to a great grade. But first, we have to understand that the first section of that analysis is the raw data. So keep in mind, hopefully in your exploration, you had 5 levels of manipulation for your independent variable and 5 levels or 5 trials for each setting of the independent variable. Your raw data, you're just simply measuring the dependent variable in each trial. You're doing that 5 times, and you just put it all in the data table with uncertainties where they're in like the overall category, like in the independent variable section and the dependent variable section. And just make sure you also have maybe some sections where you're collecting the consistency of the data. Say if you're, for example, if you're controlling temperature, you should probably have at least reference some trials where you're like, yeah, in each setting I measured the temperature and I recorded it as well, maybe in this side table, just so you make sure you get that mark as well.
Okay, but Mr. Donhow, after looking at the requirements, I also noticed that you have to keep track of qualitative observations and data. How would you recommend doing this?
So qualitative data can take many forms. In the score sheet, you might notice that they comment on maps, sketches, photos with annotations. I recommend photos with annotations, and to explain what exactly you're doing here, you're basically, supposedly you're conducting the experiment, right? So you take pictures of the experiment, and then you sort of just draw arrows and lines that and then explain certain observations you've made about the experiment. This can take many different forms depending on your research question. Just state like anything you think that is important that happened during the experiment that isn't necessarily reflected in the quantitative data. And as long as you have that, as long as you have a table for your qualitative observations, you hopefully should get the point for this box.
Mr. Donhow, can I show my face in the photos to try to appeal to the evaluators?
Nope. You are not allowed to have your face in any photos. If you do happen to have any identifying feature in the photo, just make sure you black it out and maybe edit out the photo a little bit so you're just having the photo be of something related to the experiment.
All right, the raw data seems simple enough, but how do I complete the data processing, and what are the next steps in this entire thing?
So once again, hopefully the teacher has provided you with some outline, some Google document that really kind of guides you through how you conduct the data processing, because the data processing will look very different depending on your Bio IA. Just to be simple, the data processing is basically you're taking your raw data and then you're making it into more broad statistical values that kind of help you to investigate and make statements about your research question. So the main thing is, if you're having 5 levels of manipulation for your independent variable and you have 5 trials for each dependent variable, basically you just have to, well, once again, I would recommend using the template sheet for this one, but for your investigation, I would recommend using the mean. The mean is just the average value for all, across all trials for one specific manipulation of a setting, and also the standard deviation. You should include sample calculations for each of these statistics, as well as just a broad formula. And I would recommend doing the actual calculations, like finding the mean value for each setting in Google Sheets. Hopefully your teacher showed you how to do this in class, but basically just go to Google Sheets, you put your data table in there, you have just in one of the boxes you put equal to the mean, you highlight all the required values, and then boom, you have the required statistical value. I also recommend analyzing your skew. The skew value is something that actually has a very complicated formula, but I recommend once again using Google Sheets for this. I recommend, since skew is more complicated than calculating the mean or the standard deviation, I recommend you don't actually have the sample formula for the skew, but you just kind of take a picture or a screenshot and then cut down to emphasize that you found the skew in Google Sheets. The skew value will kind of tell you about how much is your data skewed to one side or the other. If you have a negative skew value, it means it's kind of skewed to be lower than the mean. If it's positive, it means it's skewed to be higher than the mean. And and so the skew analysis depends on what value your skew is. If it's below -1 or above 1, it's highly skewed, which means there's a lot of inconsistency in your data, or it's all very far to one side of the mean. In that case, I would recommend using the median instead of the mean, and I would recommend consulting with the teacher on how exactly you go about the analysis with the median.
But also, I was looking at the scoring data, and I was wondering about preliminary data processing. Is that needed?
Yeah, so preliminary data calculations are basically just if you're doing something that involves the rate of reaction or rate of change of something, you just do the preliminary calculations to take your raw data and then turn it into a value that represents the rate. So you're doing the rate of germination. So the rate of germination would be how many seeds have germinated from the initial starting point, and then divided by the amount of days. So you have amount of seeds germinated per day. So if I was doing preliminary data calculations for you, I would just do that calculation for the mean value for each one, and then you should have your preliminary calculation.
Awesome. Once you have all the data collected and you processed it, what's the next step? Are we done?
Not quite. We still do have the impact of uncertainty and statistical significance left, but also I would just recommend going back and understanding that when you do all these calculations with your mean, with your standard deviation, make sure that you're also justifying why you're doing them. If you're doing the mean, just say that this is a way that you kind of summarize your results from a specific setting. If you're doing the standard deviation, you can say, well, this will serve as my error bars for the graph that I will have later in my section. Make sure that part of the reason why many students get tripped up and lose points is because they're not really going over and justifying every step in the data analysis section. And of course, also just make sure that all the calculations are correct as well.
One thing my teacher told me that was helpful is the data processing is more of a narrative, and instead of just like a bullet point list, so you have to explain every single thing, and that will give you full marks in that section. So Mr. Donhow, we've been collecting all these uncertainty values. What do those actually mean in the significance of IA?
So the uncertainty is basically a measurement of how inaccurate a measurement might be. So remember, back in the exploration, hopefully you collected some values on the uncertainty for your responding variable, and this uncertainty should be kept track throughout your data analysis, and make sure that you're commenting on really how it might impact your interpretation of the data. And also, once again, just like you justified the usage of values like the mean or standard deviation, make sure you're also justifying why your uncertainty is the way that it is, and also just comment on how it might be impacting the interpretation of your data throughout the analysis section.
Is there any way to ensure I get all the marks on that section?
Yeah, absolutely. So once again, just make sure you're justifying uncertainty throughout. Make sure you have the correct uncertainty for raw measurements. Hopefully that should be good, because whatever way you're finding the uncertainty in the first place should be the correct way of doing that. And make sure you maintain the correct number of digits throughout. I know I personally struggled with this, but essentially, if you're for your rate of germination, for example, Mr. Brown, you're having 2 digits for each trial. So that means all your statistical values should also be 2 digits. The mean should be 2 digits. The standard deviation should be 2 digits. Things like skew don't matter too much, but I recommend just rounding those to 2 digits as well, just so you make sure you have this mark and make sure that you're getting all the points for the impact of uncertainty.
All right.
And also, once again, this will come more into play when we're talking about the visual representation of the data through the graph later, but will discuss how the size of the uncertainties will impact the validity of the data.
Okay, so I understand how to process data. What do you do with that processed data?
So with the process data at the end, now that you should have a mean value, a standard deviation value. Remember, by the way, if your IA is a little different, I recommend consulting with a teacher on how exactly to go about certain ideas like the box and whisker plot, for example, or range of values. But if you're doing a more traditional hands-on experiment IA and you're using values such as mean and standard deviation, you will make a graph. The graph, in your case, Mr. Brown, will be a scatter plot as you're trying to represent how the value of the dependent variable changes alongside the independent variable. The independent variable will be the x-axis and the dependent variable will be the y-axis, which means the x-axis in Mr. Brown's case will be the citric acid concentration, and the dependent variable will be the rate of germination, or the amount of seeds germinated per day on the y-axis. and you should plot the mean values. You should have the standard deviation for each value, and I recommend making this in Google Sheets because it's pretty intuitive and easy to do in there. I recommend for each error bar, you put the standard deviation respective to that specific mean value, including error bars for each mean value that you plotted on the graph. And then I recommend, this will be more important if you're doing a Pearson correlation test, but also just have a trend line as well, but depending on whatever pattern you're expected to see among your data.
Okay, you mentioned the Pearson's correlation test. What actually is that, and what's the next step once you finish the graph?
So that's a reference to a statistical test that you may be conducting in your bio-IA. Once again, the statistical significance test varies very widely depending on what type of bio-IA you're conducting. There's a bunch of ways to determine which one you have. We have some ways of determining which statistical test you should take in the document linked below. But in Ryland's case, since he has five, since I have 5 manipulations of the independent variable and a bunch of manipulations of the, and we measure how it impacts the dependent variable. And it's a linear trend line that is non-monotonic, which is just like sometimes however x changes, y always changes in the same pattern. Then you will choose the Pearson correlation coefficient test. Of course, there's some different tests as well. You might use the ANOVA test, you might use the t-test, but in all these tests, you are basically finding a certain value, and then you're comparing that value with a critical value at a certain probability level to find if the result is significant or not. If it's significant, you accept the alternative hypothesis. If it's not significant, then you accept the null hypothesis. That's the main point of the statistical significance test.
Okay, I understand the test, but how do I incorporate it into the IA?
So in Ryland's case, since he's doing a Pearson correlation coefficient test, and he should use an online calculator. We have one linked in the description, but just select whatever statistical test you're doing, input your values, and then you should calculate a specific value. In the Pearson correlation coefficient test, it would be the R value, and for the R value, it does most of the calculations and hard work for you. But basically, you should select the probability level of 0.05, just because that's typically used for most bio-IAs. You then, kind of calculate using the calculator all the required values. You put it all in a data table, and then you state whether or not the value was found to be significant or not. So in Ryland's case, he should have the R value since he's doing the Pearson correlation coefficient test. He should have the sample size and also just another necessary value that might be important for the Pearson correlation coefficient test.
Okay. What's the point of the correlation test and the graph? Like, why do we need this in RIA?
So essentially what they're useful for is they're useful for analyzing your data, and you're going to incorporate them into your conclusion. The significance test is determining the extent to which you're accepting your hypothesis. If it is significant, then it seems that there is a relationship between the dependent and independent variable, and the graph is basically just showing what that relationship might be. If it's, for example, rate of germination versus rate of citric acid should be a negative trend line, as citric acid, which is our herbicide, should increase, the rate of germination should decrease. The graph is basically telling you what that relationship between the rate of germination and citric acid is. The statistical significance test is just telling you if it's significant enough to accept that the correlation might be significant.
All right. Should we move on to the conclusion?